You see sent some instructions around last week about installing Ubuntu. So one of the main reasons we opt to use Linux instead of Windows is that a lot of bioinformatic tools are built for use on a server. So all of these servers, they run some sort of flavor of Linux. So if you guys have installed Ubuntu or you're using a Mac, then feel free to click along at the same time as me. We'll try and do this together. So a little bit about bioinformatics. It's a very heavily interdisciplinary field. Lots of different experts come together to try and answer specific questions. I mean, specifically, it's some sort of combination of biological sciences, computer sciences, mathematics, statistics, all sorts of different things. So you might often hear that bioinformatics has a bioinformatics pipeline. Essentially what that is, it's just a load of individual tools which have been strung together, which will do a specific thing. So say for instance, you might have an automatic pipeline which takes your reads, builds them into an alignment, looks at drug resistance, and then outputs something nicely at the end. But unfortunately, it's quite difficult to string all those things together. So what we'll do is we'll go through some of these tools individually and then hopefully learn what's going on. So this is what sequence data looks like on the right-hand side. It's just a load of strings of nucleotides, so A's, C's, T's, and G's. This is what sequences usually look like, but you can also have them in amino acids instead. Essentially, when you run DNA through a sequencer, you get lots of different fragments. These short reads, they can be anything from 100 to 250, 300 base pairs long. These are called short reads, and this is what Illumina gives you. There's also different sequencing methods, such as PacBio or the Oxford Nanopore, which can give you much, much longer reads. Unfortunately, none of these methods are perfect, but we use Illumina, so we'll talk about Illumina today. So once you put your DNA through the sequencer, you get what we call raw reads, and these come in a forwards and a reverse direction. So when you download the files, you usually have one file which is called R1 or read1, which is the forward direction, and then R2, which is the reverse direction. And here we have a very basic sort of bioinformatics pipeline. So this is not necessarily specific to what we do on PANGEA, but this is, you know, it's good to learn so that you have some sort of idea of what you can do. So we start with downloading the forward and reverse reads, and these are called FASTQ files. We'll show you an example of what that looks like just in a minute. And then the first thing you want to do is to see how good your reads actually are. So we can do this using a program called FastQC. Once we've determined that the reads are nice quality, if we want to do anything like trimming them or anything like that, we can do that before we start to do alignments. We'll go through all this as we go along. Once we're happy that they look nice, we can then map the reads to a reference sequence. So we take all of our short reads and then we overlap and build one long continuous whole genome. And then a lot of downstream applications, they need what's called a sorted BAM file. So we can use another tool called SAMTools to sort these. So instead of having random, strings, what it does is it sorts them into nice little columns. And again, we'll see some examples of these further down. Once you've made your BAM files, these are not actually readable by humans. It's just a load of code. So we can use a BAM viewer. One that's quite well recommended is called IGV. I think it stands for Integrative Genome Viewer or something like that. There's some examples further down again. Once we've built our BAM file, we can then do another quality control check to make sure that the BAM file makes sense and it's good for downstream analyses. And then we can decide what we actually want to do with the alignment afterwards. So bioinformatic tools, I touched on this just as I was giving the introduction, but as I mentioned, a lot of these tools, they're quite computationally expensive, so you wouldn't really want to be doing them on a laptop. What you'd do is you'd send them to a cluster computer, which runs some flavor of Linux, usually something called Red Hat. All of these tools, they're basically just lines and lines of code. You don't install them in a traditional sense. You can install them using one of these installers. So there's two different types which are available on Linux machines and Macs as well. One is called Homebrew. The other one is called Condor. Homebrew is its own thing. It used to be called Linux Brew on a Linux and Homebrew on a Mac, but I think it's been all pushed together, so they're all called Homebrew now. Condor is short for Anaconda or Mini Condor, depending on which different flavor you want to use. They both kind of do the same things. It's just that some programs might be available on Homebrew, some might only be available on Condor, and so on and so forth. But as I mentioned, this tutorial is going to be primarily for Linux users. So if you followed the instructions last week and you've already managed to install Linux and you've got it booted up, feel free to click along, like I said. It's really important that you note that all of the usage commands, all of the installation commands, they're specific to Linux. So if you type these into a Mac, they won't do anything. It won't understand it. However, once you've actually installed the software, so things like SAMtools, IGV, VCF tools, etc., to actually use them, it's exactly the same on a Linux machine as it is on a Mac. So installation is different, but usage is the same. So you can follow along if you have these installed on a Mac. So, like I mentioned, all of these things are just lines and lines of code. So what we're going to do is use Terminal, which is a program in Linux and in Mac. So if we switch over to our Ubuntu instance on the left-hand side and we open all the applications down in the bottom left, you can see these are all the things that come pre-installed with Ubuntu. We just want to click down here and find our terminal. Now what I find is really useful is if you put this terminal over to the side here, so you can just drag and drop it to your favorites, or you can right-click it and you can click Add to Favorites, and then it comes up here. Just let me double check before I continue. Can everybody see what I'm doing on the left-hand side of the screen?
Yes, I can see.
You can see. Okay, that's great. Cool.
Yeah.
So we open up our terminal, and this is in Ubuntu. So we've got it pinned to the side here. We've got our terminal open, and now we can start doing things in here. So here's an example of both the terminals. On the right-hand side, this is what the one on the Mac looks like, and then on the left, it has this hideous purple weird color. I'm actually running this on my Mac in a virtual machine, so it's got like a long name here, but usually once you've installed Ubuntu, it will say something like, Steve at Ubuntu, da 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 da. This doesn't matter, it's just the username. So to get used to Linux or Unix, all of these commands that you can see here are very, very basic sort of commands which can help you navigate the terminal. So one of the most important things is ls, which stands for list. So every time you come into a directory, you want to type in ls, and then it shows you everything that's inside your directory. So for most of this tutorial, we'll be in the downloads directory. So to change directory, you literally type in cd, which stands for change directory, and then space, and then you type in whichever folder that you want to go into. Now a really nifty trick is to start typing. So for instance, I'm going to downloads. I can start typing cd, and then I can type in DOW. And then if you hit the tab key, it will auto-complete. If you've got lots and lots of files, it's quite easy to auto-complete, and then you don't have to type everything out perfectly every time. So we're typing in cd downloads / and then we hit enter, and you can see now that we've entered the downloads directory. So again, it almost becomes muscle memory. As soon as you come into a directory, you type in ls, and then you can see what's inside the directory. Well, I've gone ahead and I've got some example sequences, and I've got 2 of the fastq files, which are the raw forward and reverse reads already here in my downloads folder. So a couple of other things in here. To keep everything nice and tidy, you can type in mkdir, which stands for make directory. So for instance, I want to make a folder to put these 2 raw reads in. I can type in, say for instance, mkdir, and then I can type in, say for instance, raw. And then once again, we type in ls, and we can see now that in blue here, I've made a folder called raw. Now, if I want to move these raw files into this directory now, I can use the mv or move command. So what I can do is I can type in mv and then 14683, and then I can hit tab to autocomplete. And you can see it's only auto-completed as far as _1, _2, _0. And now that's because I could end that with _1.fastq, or I could end that with _2.fastq. So in this time I want to move the _1.fastq, so I can just type the rest of that in. And then if I want to move it into the folder, I can type in raw and then /. So this command is saying that I want to move this file here into this directory, and then I can hit enter. And if you type in ls once again, you can see now that one of the files has disappeared. And if we remember, if we want to go into a directory, we can type in cd and then have a look inside raw and then type in ls, and we can see that the file's now moved inside this folder. Now the mv command can also rename a file. So say for instance, because we're going to be using this file quite a lot, we can change the name to something a lot shorter so we don't have to keep typing it in every time. So we can type in the name of this file, and then say for instance we might just want to call it r1.fastq.gz. So here it's saying we want to move this file to this file instead. I mentioned that the fastq is the raw output. GZ means that it's been compressed with something called gunzip. A lot of programs will read it with the .gz, so you can leave gz as it is. So we hit enter, and then we'll go ls again, and we can see now that I've renamed my file r1.fastq.gz. So what we'll do is we'll just move this back to the directory above. Another neat trick, you can see here that I've typed in here cd space .. basically means go up one directory. So here I'm going to move r1.fastq.gz up one directory and then hit enter. And now just to double check, go ls again, and we can see there's now nothing in this directory. So we'll go up one directory, and now here you can see here's my r1.fastq.gz, and here's my other two directories. So we'll make this one shorter as well, so we'll just type in mv 14683, and you can see it's auto completed the whole thing now because there's no other possibility, it will only be that. And we'll call this one r1.fastq.gz and then hit enter. Overriding mode. This is something to do with permissions, so we can just press yes, type in y, which means yes. Oh, okay, we've lost the file now. Ugh. The problem with terminal is if you make a mistake, you can't get it back. So what we'll do is I just need to move the file back again. Okay, we'll just leave them as they are for now. So here we have our forward read, our reverse read. We'll just remove that one so we don't get confused. Yes. Okay, so there's a quick tutorial on basic Linux commands. So to actually install Homebrew, we can just follow exactly what's written down here. So sudo means you're giving your terminal elevated permissions. So a lot of things that you do in Ubuntu, it's like right-clicking and clicking run as administrator on a Windows PC. So if you just type in apt install, it doesn't necessarily have the right permissions to make that installation. So we type in sudo apt install. So what we'll do is we can just copy and paste this into our terminal and then hit enter. And then every time you do sudo, it'll ask you for your password. And that's just to make sure you want to do what you want to do. So you'll type in your password. As you're typing, it won't show it. It's all hidden. So just type it in and hit enter. I've installed this already, so it just says this. But what it will do is it'll come up with a long list of stuff that's installing. You just press yes or whatever to install it. And then afterwards, what you have to do is this is installed to a random directory, the bin directory. To access it from anywhere, you have to add it to what's called your path. So here on the right-hand side, you can see that there's all these different pieces of information. You just copy and paste these again, put them into your terminal, paste, and then hit enter. And then what that does is, if you go echo $path, you can see now it's added all of these different things to my path. So now every time I type in brew, which is the command to open Homebrew, I can be in any folder that I want, and it will, it will find it. So here you can see that if you type in brew, it will open brew anywhere. If I go back into the downloads directory, I type in brew, you can now access it from anywhere at all. Okay. So that's one of the ones we can install. Another one is called Condor. To install this, it's a bit different. We use this command wget, or widget, which some people call it. Widget basically it's like a code downloader. So this is the web address to where this file is located on the internet. So we open our terminal. We're in the downloads folder. We type in widget this. We hit paste. We hit enter, and you can see it's now downloading this file. It's all downloaded now. So if we type in ls, we can see now it's here. That's the file there. So now to install this, we run this. So .sh means that it's a script file. So it has loads of instructions inside this file to install everything and put everything in the right directory. So to install a script file, you type in bash. So we can type in bash, I can start typing mini, and then I can hit tab and it'll autocomplete, hit enter, and then you can see down on the left hand side it has some instructions. You just follow these. So it says press enter, so hit enter. It gives you loads of gibberish about licenses and whatnot. You just keep hitting enter to skip through all these, and then it'll say, do you accept these terms? You hit yes, and then it'll say it's going to install it into this directory. And you can change this if you want, but I'd advise you just leave it where it is. This will say something like home, and then your name will be here, and then it will be inside this folder. So hit enter to install. I've already installed this, so it said it's already installed, but you just hit enter, and then I think you have to type in Y a few times. And then it's important now that because we've installed these two things, we have to close and reopen the terminal so that it can load it. So just close your terminal, and then go and open it again. And to check that it's installed, you can just type in Condor. And because the script file adds it to the path automatically, you should be able to see it anywhere. So you can open this wherever you are, whichever folder. So one more thing we have to install is a Java environment. So Java is just a language, programming language. It's just quite common. A lot of bioinformatics tools are written in Java. Ubuntu does come with a version of Java, but not the development program that you need to run these software. So this one's very easy to install. You just type in this command into your terminal. This one takes quite a long time, so I went ahead and installed this in advance. So mine's quite short here, but it usually takes 5, 10, 15 minutes depending on your internet connection. So you can make sure this is installed properly by typing in Java space -version. If it's properly installed, you'll see here, this is the version of Java that I have installed here. So this is installed Java 11. There's different flavors, Java 8, 9, 10, 11, so on and so forth. Some programs use Java 8, some use Java 10. You find out by using them. Okay, so now that we have everything installed, we can start doing some analyses. So we already had a look at the raw reads. So our raw reads here are the FASTQ files. This is basically what they look like. The forward read goes from a 5' to a 3', and then read 2 is basically the reverse upside down. So it kind of looks like this. So your _1 or R1 file is your forward read, and then your _2 is your reverse read. So this is what a FASTQ file looks like. It's just a plain text file. So we'll look inside this one. So if you want to ever open a file in Ubuntu, there's lots of different programs which are pre-installed. The easiest one is just called less. So you can type in less and then the name of the file. So here we'll look at the forward read. So if we open this up, this is what it looks like. So this top line is just the name of the read. This second line is the sequence itself. So I mentioned all these reads are short reads, so that small sequence that you can see highlighted here, that's one read. The third line is a plus symbol. It can have some sort of duplication of the read name here. It doesn't always. Here it's just empty. And then these crazy characters here, these are quality scores. They're written in something called ASCII, which is like a computer code. So you can Google these, but basically they go from something like, I think one symbol is lowest quality, and then it goes all the way to, I think maybe Z or something, which is the highest quality. So each of these matches each of these letters. So say, for instance, this C here, that's the quality score for that C, and so on and so forth. So that's what a FASTQ file looks like. So what we're going to do is we're going to have a look at the quality of these FASTQ files. I mentioned earlier that we're using the Illumina platform, which gives you loads and loads and loads of small sequences. We need to know if those sequences are believable, if they're good, or if they're not good. So what we're going to do is install a program called fastqc. So we can do this by typing brew install fastqc into our terminal. So fastqc, hit enter. Mine's already installed, but yours will say something like updating Homebrew, and then it will pull a list of all the things that you can install with Homebrew, and then it takes maybe 5 or 10 minutes, maybe 5 minutes even less to install. Once it's installed, it goes straight into your path, so you don't have to find the folder. You can literally just type in fastqc, and it will open. So fastqc, you can either use it with a GUI, which is where you point and click. So if you just type in, oops, it didn't work that time. Okay, it's not working. Sometimes it doesn't work properly. If it doesn't, then you can just use this command instead. Ubuntu's quite good, it'll just tell you here. So it's saying that it can't find it, so we're going to install it with apt install instead. So you can just do this. So you see here it's asking for some extra dependencies. So where it says, do you want to continue? You can just type in yes. And then it will write loads of random gibberish that we don't have to worry about. Okay, so now hopefully if we type in fastqc, it should work. Okay, here we go. So if we type in fastqc, it's opened here. So as I mentioned, you can use it as a GUI, which is where you can point and click. A better way to do it is to just type in fastqc and then all the names of your reads. So you remember our reads are called 14683 underscore da 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 da. So fingers crossed, if we type in fastqc 14683, we type this in here. So we're going to call fastqc and then give it the read. If you hit enter, hopefully it will run. Yeah, so that's promising. So what it's doing now, it does the analysis and then it writes a HTML file with all the analysis. So we wait for this to finish. It shouldn't take that long. There we go. So you see it's finished now because your cursor's come back up. What it does is it outputs a zip file, which is this one in here. Inside there, it's just got some individual metrics, or you can just open the .html file, which is more advisable. So you can either go into your files and open it manually by opening your files, going into downloads, and then double-clicking this. Or if you're on a server, you can't do this. So what you can do is you can type in sensible-browser and then the name of that HTML file, which is called _fastqc.html. And then you hit enter and it will open the HTML. So this is what it looks like. We'll go through some of these together. So looking at basic statistics, it tells you the file name you've put in, it tells you what you've used, which is Illumina, it tells you the total number of short sequences that it's made, the average sequence length, and the GC content. So a good sequence, you want to have 50-50 GC to AT. It says it down here on the left-hand side though. So moving over to the right-hand side, we look at assessing the read quality. So it uses something called a Fred score, which is a quality score. I want to say it was originally made by Sanger. Don't quote me, I think it's made by Sanger. Essentially, a Fred score of, and let me just double check this before I give you wrong information. The Fred scores relate to, yeah, here we go. Okay, so a Fred score of 20 means that the base that's been called is 99% accurate. And then a Fred score of 30 means that the base called is 99.9% accurate. And then a Fred score of 40 means that it's 99.99% accurate. So here you can see the first load of sequences here are all above 28. So we're happy with all of these sequences. And then the further you get towards the end, the worse they seem to get. So anything below 20, we might consider trimming away because these are all low quality, and we're only sort of 99% sure that it could be these. And 1% error rate is quite high, so we might want to trim these away. If we go down a little bit on the HTML, it has all sorts of different graphs and figures. Not all of them are so important. We'll just go through the important ones. So here we can see the per sequence quality score. This shows the average quality score of all the sequences. So you have a number of sequences on the left-hand side and the Fred score on the bottom. So you can see that the line is basically flat up until we get to 28, 29. This is telling us that almost none of the sequences are really, 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 really bad. When you have a quality score that's less than 27, you'll get a big warning here. And if the average is less than 20, so say for instance your peak sort of peaks, here at 20 instead of at 35, you'd get basically what's called a failure. So it would, instead of having a nice green tick here, it'd have a nasty X. It would basically say that it doesn't trust these reads at all. So this is what a nice read looks like. The majority of the reads, they fall here with Fred scores of above 30. So next we want to have a look at the sequence duplication levels. You'd expect that if you're getting something like 150,000 sequences, or short reads, per sample, you'd expect them to be quite diverse. So you wouldn't expect to be so many duplicates. If there is lots and lots of duplicates, that will tell you a few different things. It might tell you that there's some contamination, the library hasn't worked very well, or there's PCR duplicates. PCR duplicates are not great. They affect the downstream analyses. So here we can see, this is an actual example. So the blue line is what we actually have. So you can see that 25% of our sequences were only seen one time, and so on and so forth. This is percentage on the left, and then on the bottom is the duplication level. So you can see that maybe that 8% of our sequences have been duplicated more than 10 times. And here, which is a bit worrying, about 10% of our sequences have been duplicated more than 100 times. So if we deduplicated the sequences, it should look like the red line. So you can see that if we run a deduplication software, then 65% of our sequences should be only seen one time, and about 15% of sequences should only be seen twice, and so on and so forth. So blue line is what we actually have, red line is what we want to have. A bit further down as well, it gives you the sequences that are overrepresented. So any sequence that you see here makes up more than 0.1% of the total of the reads. So FastQC has an inbuilt database which is comprised of sort of contaminants, known contaminants. So you can see here down the right hand side it says no hit. So it's telling us that this is not contaminants, it's just something else has happened. So this sequence here has been seen 7,271 times out of 261,568 times. So we're not sure what this is, but we don't really want so many overrepresented sequences, so we might consider taking these out. Another quite important thing is to look at the adapter content. So when you do Illumina sequencing, adapters are added to the DNA or the RNA, and then they're used to actually do the sequencing. FastQC gives you a report of all the adapters, and then this is the cumulative amount of adapter that it's seen in the sequences. Ideally, you want to remove the adapters because, again, they can skew your downstream analyses. So if you have loads of adapter sequences and you try to align to a reference, it doesn't like it so much. So you can see that here we have quite a lot of the universal adapter. So there's a couple of programs which will help us clean these reads up. One that we recommend is called Trimmomatic. We'll go through that in just a bit. So if you do have any of these issues, so say for instance on the left-hand side, this is a real sequence that we use as part of PANGEA. This is just the raw sequence. So it has quite a high duplication level, it's got quite a lot of overrepresented sequences, and it has loads of adapter. So we will fix all of these things before we start to do an alignment. So we'll do this using the program called Trimmomatic. So to install Trimmomatic, we go back to our terminal. We make sure that we're in our downloads directory, which we are, you can see down here. And then we'll use widget again. So you can just copy and paste this into your terminal. Hit enter, and you can see that it's downloading the Trimmomatic zip file. Type in ls to have a look what's in the directory, and you can see that there's a zip file here. So literally just keep following this down. Next we need to unzip it. So we'll type in unzip Trimmomatic, enter, and you can see on the left hand side it's unzipping this into its own folder, which is now here. So we'll go into that folder now by typing change directory, go into that folder and have a look what's inside. And you can see that there's a jar file. So in order to open these jar files, you need to have the Java environment installed. That's why we started off with that. It's quite important. If you don't have Java installed properly, you won't be able to do anything with this jar file. It will just give you an error. Another thing we need to do is to move the adapters out of the adapter folder. So the Illumina adapter sequences there are copyrighted, patented. They're not meant to be freely available, but Illumina has given Trimmomatic permission to include the sequences. So what we can do is we can change directory into the adapters folder. If I can spell properly. Go ls, and you can see here's all the different types of adapters. Off the top of my head, I can't remember what each of these are for. You can have a look in the Trimmomatic manual, which is available on the Trimmomatic website if you want to know what these are. So we want to move all of these adapters to the folder above. When we open this jar file, it can find them all. So to do that, we can do move. Because we're already in the folder, this is slightly different. So if we just go back out of the folder again by going up, what we can do is we can type in move, and then adapters, and then star. Star is a wildcard, so it means all. So say for instance, I'll give you another example later on, but star means all basically. So we want to move all of everything in the adapters folder, and then we go dot. Dot means this directory. So we're gonna, without going into that folder and then coming back out of it, we're staying in this one folder. It's gonna look in this folder and it's gonna bring everything in that folder back down to this folder using this command. So we hit enter, and then we go ls, and you can see now that it's moved all of the adapter sequences to this folder. So now what we want to do is to remove the adapter sequences, we type in this long thing here. Anything you can see here in curly brackets like this is something that you should change, that is specific to your computer. So don't just copy and paste blindly, because this won't do anything, it will give you an error. So we'll go through this together. So Java means that you want to use Java. jar means that you're going to open a jar file, which is a Java program which has been packaged into a single file. And then you point it to the jar file you want to open, which is called trimmomatic.0.39.jar. And then you start to give it arguments. So PE means paired ends. And then here we have dash threads. So when you're running this on a cluster computer, they're much more powerful. You know, they might handle 64, 32 threads. The laptop that I'm running off at the minute can handle 8 threads. If you're not sure how many threads you can use, then just don't type that in, basically. But the more threads that you use, the faster it's going to be, which is why we often ship these things off to a cluster computer. So I'm using 2 at the minute. That will make it, you know, a little bit faster. And then the next one is the Fred score, which is the scores we talked about earlier up here. These quality scores. This is the Fred scores. So 33, it doesn't mean a Fred score of 33. It means something like if the score starts with 3 and ends with something. You can look at all this in the manual if you want to really understand what's going on. But yeah, we use Fred 33, and then you put in your forward read and your reverse read. These are going to be in the folder above now. So if we want to access a file that's not in the same folder that we're in, we can use the same command as before. So we can do ..// and then we can type in the name of our sequence, which is 1468_1_fastqc .gz, which you can see here. That's our forward read, and then we also want to do the reverse read, so we type in _2. So here we have paired ends, 2 threads, Fred score of 33, forward read, which is there, reverse read, which is here, and then we type in -base out, and then give it a name of whatever you want to call it. So this particular faster file is called 14683, so I might type in 14683_test. And base out basically tells it that it wants you to print the unpaired analysis out and the paired analysis out. You can type in four file names that you want, or you can just type in base out and it will do that all automatically for you. Now you can see all of this gumph at the end. I just copied and pasted this from the manual. This is the default settings, which will remove all the adapters. So we can just copy and paste that directly into our terminal. So now we have a command that's specific to my laptop. So we have Java, jar, we've pointed it towards the jar file, we've told it that this is a paired end. I want it to use 2 threads. I want it to use a Fred score of 33. I've given it the forward read, which is there. I've given it the reverse read, which is here. And I've told it that I want it to output files called these. And then this is all the different steps that's going to run. Now, if that's all correct, what we do is we cross our fingers, we hit enter, and hopefully it does something. Main thread. Okay, this is giving me an error, so it's telling me that this file doesn't exist. Maybe I typed it in wrong. We'll just go and have a quick look. Okay, yeah, I've missed it. Maybe fastq.gz. Okay, you give it a file. You give it 2 files basically, and it outputs 4 files. It's telling me that it's not finding this file. It's probably because I'm using this inside a Mac, which you shouldn't be doing. But essentially that will give you 4 files out. It will give you whatever you've typed in here called example 1p, which is the forward paired read, and example 2p, which is the reverse read. So these 2 files will have all the adapters removed. So if we go back up here, if you rerun fastqc, you'll see that this is all gone now. There'll be no adapter sequence left in those forward and reverse reads. So once you have those reads which have been cleaned of adapters, you can also change these arguments to remove the duplicated sequences. They're all specific to the reads themselves, so you have to read the manual to have a look and see how to do that. Yeah, I can help you if you have any examples which you want to send to me. We can look at them together. But essentially you get 4 files out which have been cleaned. So once you're happy that they've been cleaned, you can then start to map these. Now there's 2 different ways you can do that. You can do what's called de novo assembly, which is where it doesn't have any framework to work off of. It will literally look at all those individual reads and it will try to make the best long genome it can. That's useful if you don't have a reference sequence that you want to map to. With HIV, we have lots and lots and lots of reference sequences, so we do what's called mapping to reference. There's lots of different mapping tools that you can use. The one that I like to use is called BWA, which stands for Burrows-Wheeler Aligner. There's lots of other ones. There's BBMap, there's Bowtie, there's some online tools. You use whatever works for you. But this one's nice and quick, so if you're not using a cluster or a server, it will do it quite quickly on your laptop. So what we'll do is we'll go back out of this folder, go back into our downloads folder where we have our reads, and then we'll type in sudo apt install bwa to install bwa. Type in your password to give it permission. It will install. I've installed it already, so it's saying this. Yours will be a bit longer. And then we double check that it's working by typing in bwa, and here you can see it's found it. So this is what you'll type in to do whatever you want with it. So we need to download a reference genome. I'm just going to use HXB2 because it's easy to find. So what we can do is we go to Firefox, which is the standard one in Ubuntu. Just type in HXB2 reference genome. Go to NCBI and just download the file. So here we just we click on faster. And then you go on to send to, choose destination file, make sure this says faster, and then if you go create file, make sure you save it, because if you open it, you can't do anything with it, you have to save the file. And then press OK. Here you can see it's finished, so that will have gone into my downloads folder, sorry. So just double check it's there by typing in ls. It's called sequence.faster because I didn't call it anything else. We'll change the name so we know what it is. So we'll do move sequence.faster. We'll call it hxb2.faster. ls. And you can see my hxb2.faster. If you want to have a look inside it, you can do less hxb2.faster. Here you can see human immunodeficiency virus hxb2. There's my sequence. To get out of less, you just type in Q and it will quit. Okay. So before we do anything with the reference file, we have to do what's called an index. So BWA will handle all this for you. You just type in BWA and then the name of your reference file. So we've called it hxb2.faster. So we do var index hxb2.faster, hit enter. And you can see it's very quick. It's now made loads of index files. So if you type in ls, you can see .amb, .ann. I don't know what any of these are. It's the index files. You don't have to worry as long as you get this. All of this stuff here. If you get an error, then you've done something wrong, or you've not downloaded a faster, or you've given it a wrong name or something like that. But once you've had all these indexed, you can then start to give your reads to BWA, and it will make you an alignment file. So BWA has lots of different settings. We'll use something called BWA mem. You can read about that on Google if you want. But essentially, we type in BWA mem. You then give it your reference genome, which is the HXP2.FASTA, and then go space. And then you want to give it your forward read. .FASTQ, and then your reverse read directly afterwards. So you can see here it's BWA space mem, give it your reference genome, which is that one, and then give it your forward read, which is this one, and your reverse read, which is this one. And then you need to tell it where to output. So you go -O, and then say for instance we'll call it 14683.sam. Hit enter, and then it will start to do the alignment. It shouldn't take so long if you've got a fast computer. If you have a slow computer, it might take a little longer. But in the tests that I've done, it didn't take so long. Whilst it's running, does anybody have any questions at all? No questions? Everyone happy? Everybody's happy. I'm also conscious it's getting on in time. I don't know if anybody wants to have a break or if we want to keep going. We're about halfway, I'd say. Everybody's okay? Oh, I'm just seeing loads of things in the chat. That's good. How does Illumina determine the quality of a base? Off the top of my head, not entirely sure. I just look at the downstream. I don't actually do sequencing myself. Illumina does have quite a lot of good information though, so you can read up, or I can give you a link to the file afterwards. Okay, nobody wants to take a break, so we'll keep going. So you can see that this is finished now, and it's output a SAM file, which is an alignment file. So a SAM file, scroll down a little bit. It stands for Sequence Alignment Map. This is human readable. So what we can do is we can do less 14683.sam, which is the file that we've just made. And this is what the SAM file looks like. So it looks like gibberish, but it's readable. So here you have, this is called the header. So this basically tells somebody which aligner they've used. So we've used WAMEM, we've used this version of WAMEM, 0.717. This is the reference that we used. This is the forward read. This is the reverse read. So this is the output. So this tells, so for instance, I send you this SAM file, you can see exactly how I've made it. And then we have lots of different things here. So if you can open this in a text file. It's in columns. So you can see here, the first column is the read name, which is this long MS8 da 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 da. You then have something called a SAM flag. There's all sorts of flags. There's a flag reader online if you're really interested. I don't know what any of them mean off the top of my head. The only important one that I need to know is a SAM flag of 4, which means that it's unmapped. The next one is the name of the config, which is, it didn't print the name. And then the position of the base, mapping quality, so on and so on and so on and so on. Okay. You don't ever really need to open a SAM file, but it's nice to see it just to understand what it looks like. To do anything with these files, we need to convert a SAM to a BAM file. So a BAM file is a binary alignment map. It's not human readable, it's all just coded gibberish. It's also quite heavily compressed. So SAM files can be quite large. They can be, 5, 6, 700 megabytes each. BAM files tend to be much, much smaller because they don't contain all that information in plain text. So to work with SAM files and BAM files, we use this program called SAMTools. So we go across to here, the terminal, and we can install it. So sudo apt install SAMTools, hit enter, it will install it. I've already got it installed, so mine's shorter, yours will take a bit longer. And then type in SAMTools just to make sure that it's working okay. You can see on the left-hand side it's working because it's given us all these options. Now to change a SAM file to a BAM file, we use this command SAMTools view, which is how you can look at a SAM or a BAM file. -H means also print the header. -B means, I wrote this down. -B means it'll output a BAM file, and then -S is the input file. So we've done SAMTools view -H, -B, -S. It's also important that the first two are lowercase and the S is uppercase. These are specific, so you have to have lowercase or uppercase. The lowercase letters might mean something different, so you can just copy and paste the example. So we've done SAMTools view. We'll give it the SAM file we've made, which is called 14683.sam. And then we use greater than sign, which means output. And we'll call it, say, for instance, 14683.bam. Hit enter. It will take a little while. It compresses, recodes. It does some stuff to it, basically. Shouldn't take so long. So you can see it's finished. So if we type in ls, you can now see that we have our BAM file, which is this one here. Now I'll just show you what a BAM file looks like. It's complete nonsense. It's not human readable. Yes. So you can see it's all just, lines of gibberish. It doesn't mean anything to us. So that's why we first output in the SAM file if you want to check it for errors, if you want to check it for anything, and then we convert to a BAM file, which is what the computer then uses downstream. Okay, so I didn't mention, when you make an alignment, you might have human reads, contaminants, all sorts of weird stuff which doesn't necessarily map to your reference genome. You can remove these if you want to make the file much smaller and to make downstream analysis quicker. We can do this by using this, this command here. So when I said that there's only one flag that I know, it's flag 4, which means that a read has not been mapped properly. So we can type in SAMTools view -B -F4 and then give it your BAM file 14683.bam greater than sign, which means output, and then call it something like mapped_14683.bam. Hit enter, and it will basically write a new file called mapped_bam. Again, it'll be much smaller because it won't have printed all of the unmapped reads. So we just press type in ls, hit enter, and we can see mapped.bam just here. So if you look inside that file with a viewer, you might see that all the reads are just here, there, and everywhere. So what we'll do is we will sort and index the file, which is required for some downstream analyses. Again, we can do all these with SAMTools, so we don't have to install anything. There's two different commands. SAMTools sort sorts the alignment. There's two different ways you can sort. You can sort by read name, which is not very helpful, or you can sort by coordinate position. So HIV is 9,700 base pairs long. All of the reads that mapped, I don't know, to like 1 to 150 will be on the left-hand side, and then it will cascade downwards. And then SAMTools index is the second command. It basically writes an index file, which is read by some programs downstream. So the first thing we'll do is do SAMTools sort. So on the left-hand side, type in samtools sort. We're going to give it the mapped BAM file that we've just made. And then we'll type in -o, which means output. And then we'll call it sorted _ 14683.bam. And then hit enter. It will run something in the background. And you can see it's finished. So we now have a sorted BAM file. And then we will index it as well by doing samtools index sorted BAM. Hit enter. It's very fast. You now have this index file, which is called .bam.by. You don't have to do anything with it. It's there and it's read by some programs. Okay, so the important part now, we want to actually have a look at the BAM file. We want to see if there's good coverage of all of the genes that we're interested in. We want to see if the BAM file makes sense. So we're going to use a viewer called IGV, which is on Windows, it's on Mac, it's on Linux, it's everywhere. On Linux, we download it by typing in this command. So we're still inside the downloads folder. We paste this and we hit enter. A note that I saw this morning, on the original PowerPoint, I think this said 2.7.0. It's been updated since I wrote the PowerPoint a few days ago, so I've changed it in this version. We will upload this copy that we're reading off of at the end of this webinar, and then you'll be able to get the right command. You don't have to use widget. You can download it using a web browser if you want. So you can go to Firefox. You can type in IGV for Linux. Go to the actual web page, and then you can click download here. You can save it. Widget and downloading it manually does the same thing. It's just, it's a bit more streamlined if you just do everything in the terminal. If you're doing it on a cluster, you won't be able to do this, obviously, which is why I've given you these instructions on the right-hand side. But anyway, once you've downloaded that file, it's still coming. Wait for it to finish. Quite cool. It gives you a little ETA, how many seconds it's going to take. So you can go make a cup of tea if it's going to take more than 5 minutes or something like that. Almost there. Okay, so we can now see that we have another zip file. This one here, IGV Linux 2.7.2.zip. Remember from previously, we have to unzip it into its own folder. So type in IGV, hit tab, it will autocomplete, hit enter, and you can see that it's written to its own folder, which is this folder here, IGV Linux 2.7.2. We change directory into that folder. We type in ls to see what's inside there. And you can see that we have these .sh files again, which is the script file. So to open this, what we want to do is type in, not bash this time because we're not installing anything, we type in sh, which means run script, igv.sh, and hit enter. And you can see that it's using the Java that you've installed earlier to now open this program. And here on the left-hand side, this is what you can see. So, IGV viewer. What you first have to do is give it the reference file that you've used to make the BAM, otherwise it won't open anything. So here you can see that it has preloaded this human HG19. What we'll do is instead, we'll click on genomes, we'll click on load genome from file, we'll go into downloads, and then we'll give it the hxb2.faster, which is the reference sequences that we used to make that BAM file earlier. So select it and then hit open. And you can see now it's loaded my my FASTA file. And this is the name of the name of the chromosome that it's loaded. So now that we've loaded this, we can now load we go File, Load from File, Downloads again. We switch it to Details, and we see which one we've modified last. And we want to give it the sorted BAM file, not the index file, the actual BAM file itself. And then we hit Open, and you can see in the corner here it says Reads Loaded. It'd take a little while to actually load all the reads. And here we can see our BAM file. This little box here, it might be a bit small. I apologize if you can't see it. You can zoom in when you do it yourself. This is a plot of coverage. So you can see at the beginning of the alignment, there's quite a lot of coverage, and then there's not so much in the middle, and then there's a little bit here and here and here. A lot of the PANGEA sequences, well, not a lot, some of the PANGEA sequences were only partial genomes. So when you actually build the BAM files, you might get a lot which cover pol, you might get a lot which cover gag, you might get a lot which don't have any envelope. You know, when you build the files, you'll see this. This is why we view the BAM files to see what we're working with. Because we've sorted it, you can see that it's all the reads are nicely stacked. So if I scroll down, they get smaller and smaller the further that you go down, because we've sorted them essentially. So here we can see that we're missing quite a large portion of the genome. So I don't know, if I wanted to do some sort of resistance analysis, I would probably not do any resistance analysis with this because it might be missing large parts of the polymerase gene, which is not useful for me. But anyway, this is just an example. So we've made our BAM file. This is how you view it. Here's another example that I used earlier. It's just very important to remember you first have to load your reference genome in here, and then you load the BAM file that you've made afterwards. So once we've had a look, you can close that down. You have to close it first, otherwise your terminal will not be usable because it will still be running stuff in the background. So make sure you close down IGV. Scroll. Once you've made your BAM file, you can then do fast QC again to look at the BAM file. It will give you the same metrics. So once you've trimmed it, once you've taken out adapter sequences, whatever, you can run fast QC on it again in the same way. So you can just type in fast QC, and then you give it your sorted, whatever the BAM file's called, which folder I'm in. Oh, sorry. Go back up to the downloads file, downloads folder, and then do fast QC sorted.bam. Hit enter, and you can see it will do the analysis of your BAM file. It will give you another HTML file. You can have a look at the report. You can decide what you want to do with it later on. Okay, so we have taken our raw reads, our forward and reverse raw reads. We have assessed them for quality by using fast QC. We have trimmed off the adapter sequences and any extra duplicate sequences that we don't want using Trimmomatic. We've then made an alignment using BWA. We've then had a look at that alignment using IGV. No, we haven't. We sorted and indexed, and then we had a look at the BAM file. We've made our BAM files. We're happy at this point. Now we need to figure out what we want to do next. So there's a couple of different downstream analyses that you can do. Some people want to make some trees. Some people want to look at drug-resistant mutations. Some people want to do ancestral reconstruction. You figure out what you want to do, and we will help you do it if you need help doing it. So when we were at the phylogenetics course in Durban in September, a lot of people were interested in making trees. If you're using Phyloscanner, if you want to use Phyloscanner, then the input is the BAM file that you've made. But if you want to make maximum likelihood trees, if you want to make beast trees, if you want to make any other sort of tree, you do this using a consensus sequence, which is the most common nucleotide at each position. PANGEA has made consensus sequences and BAMs that if you become accredited, which I'll mention in just a little bit, it's just further down, you can request data You just write a short letter of intent, basically saying what you want to do with them, what support you need, etc. Lucy can help you with that if you need some help. But yeah, PANGEA keeps consensus sequences and BAM files. But if you want to make a tree using something like RaxML, or if you want to look at the alignment of consensus sequences, you need to build what's called a multiple sequence alignment. So when you get all of your consensus sequences, I've downloaded some already. I've put it in a folder called example sequences, so we'll have a look together. You can see that I've got 5 PANGEA consensus sequences. These are South African samples. They were sequenced in 2016. We've got some metadata on them as well. What you'll want to do is to make a multiple sequence alignment, you first have to put them all into the same file. So you can do this by getting all your consensus sequences and then typing in cat, which is short for concatenate. Because there's only 5 sequences in that folder, we can just type in cat and then do the asterisk, which means all. And then we want to do the greater than sign, which means output. And I'm just going to call this, I don't know, seeks.faster. Hit enter, type in ls, and you can see I've made a new file called seeks.faster. If we just open that, we'll have a look what's inside. So we can open it using the less command. So here you can see that it's put the first sequence, well, the first sequence is there, and then the second sequence is here. Each sequence is delimited by this greater than sign. So press Q to escape. What we'll do is we'll have a look at this using a program called AliView, which is also recommended by the Los Alamos people. It's a nice program. It does alignments. You can also get it to build trees for you as well if you want to point and click. Yeah, we'll have a look together. So to download AliView, we use this command again, which is wget or widget. We make sure we're in the downloads folder, which is here. We type in the command, we hit enter. It will download this file called, AliView install.run. Just make sure it's there. It's there. And this is a slightly different file. You have to give it permissions to run. So we do this using a command called chmod. So you can just copy and paste from the PowerPoint and put it in here. We do chmod plus x, AliView install.run, hit enter, and that just gives it some additional permissions so that it can actually run. And then we type in sudo, and then we do dot forward slash. Dot forward slash on a Linux system means run this thing. So dot forward slash AliView install.run, hit enter, type in your password, hit enter, and then it will install AliView. It installs that directly into your path, so to open it you can just type in AliView, hit enter, and you can see it's opened here now. So if I make it a bit bigger so you can see, what we'll do is we will open our sequence that we've just made. So it's seeks.faster, hit open, and you can see here's our sequence. Not aligned, it's not in alignment because it's just the consensus sequence that we've added. So in order to build a tree, you have to make a multiple sequence alignment. I don't have my reference sequence in here, so we need to add it to this file. You can do that in 2 ways. You can copy and paste it directly into the program, or you can follow the instructions here. Sometimes I find it easier just to copy and paste, so we can open up our files, go to downloads, go to hxb2.faster here, and then you can copy, and then hopefully you should be able to paste. Yep, so you can see that my reference sequence has gone into here. And then what you can do is you can go align up here and go realign everything. Are you really sure you want to realign? Yes. And then depending on how fast your computer is, it will redo the alignment. Sometimes it takes a little while. Depends on how big, how many sequences, how fast your computer is, whatever. Once it says done, it's done. So you can click on the X, click OK. And then if we look across now, it's inserted all these gaps into my sequences because this is just the LTR region. And then you can see that it's nicely aligned. Always make sure you check your alignment before you start doing anything because sometimes they're not very well aligned. This uses something called muscle. There's lots of different alignment tools. Some good ones you can see on the right hand side of the screen here is Gene Cutter or HIV Align. These are on the Lionel website. HIV Align uses something called MAFT, which is just a different alignment tool. Gene Cutter is better because it looks at codons, so it takes each 3 amino acids and it tries to make proper sense of them instead of just inserting random insertions and deletions everywhere. It's also an online tool, so if you have a very, very big alignment, you send it to their server and you type in your email address, it will take about an hour to run, and then they'll send it back to you. Okay, tree building. So you have your aligned sequence here. We want to save it, otherwise there's no point in doing an alignment, so just click save. And then you can see it's just saved here as sequence.faster, wherever it is. Oh, it's an example, so yeah, it's here. So this is now aligned because we've run it through the alignment tool and we have saved it. So there's 3 different types of tree. Anybody that was at the course might remember Brian gave a talk about different types of tree. There's neighborhood joining, maximum likelihood, Bayesian trees. Depending on what you want to do, you can use different ones. I always recommend doing maximum likelihood trees. They use more complex evolutionary models, so it tends to give you a better or more accurate tree. But if you're just trying to build a tree quickly, then neighborhood joining tree, it's fast, it's quick and dirty. One of my colleagues always says. So it'll give you the general idea of what the tree looks like. But then using a maximum likelihood tree with bootstrapping probably give you the most accurate tree, I suggest. So we use a tool called RaxML to build maximum likelihood trees. You can go ahead and install this by using this command. Paste, enter. Aha, I haven't installed this. So this is what an actual install looks like on the left-hand side. That just gives you loads of gump you don't have to really worry about unless it comes up with big red warning signs. But this is finished and it's installed. RaxML is a bit confusing to use because there's so many different versions. When you install RaxML, to call it, you type in something called RaxML HPC. But there's different versions. There's RaxML HPC AVX, which is for newer processors. It's a bit faster. You can parallelize this to use different threads for building different bootstraps. The manual's like 100 pages long. You can read that in your own time. But to make sure it's installed, we just type in RaxML HPC, enter. You can see it's given me an error because I've not given it any arguments, but it's installed. If it wasn't installed, it would just say, I don't know what you're trying to ask me to do. Okay, so we'll build a tree, and then I'll show you how to manipulate it, and then we'll probably stop there. So RaxML HPC -F means, -F is what you want it to do. So -FA is like a complete, does everything in one. So it will do bootstrap searches, maximum likelihood searches, find the most likely tree, and then add bootstrap values to it. So it's like an all-in-one sort of thing. So -FA and then -M. M is the model that you give it. So you can build trees using amino acids, or you can build using nucleotides. We're using nucleotides at the minute, so we'll use something called GTI gamma, which is kind of like the default one that's in all of the tutorials. And then -P is a random seed. if you want to, every time if you give it the same seed each time, it should give you the same tree each time. So it's useful for debugging. I give it a random seed each time, and then get different trees each time. So you can type in $random in curly brackets. That means it'll give it a random number each time. -x is the number of bootstrapping seeds. So because we've done -fa, we need to give it two different types of seed. So again, we can give it a random seed, and then I don't know what we call it, pound or hashtag is the number of bootstraps that you want it to do. If you want to make a fast tree, then you can do 10 to 100 bootstraps. Publication quality is anything more than 500. So between 500 and 1,000 I usually do if I'm making proper trees. Just because I want to show you guys really quickly, we'll just do 10. And then -s is the name of the alignment file that we've made. So we called it seeks.faster, so put seeks.faster, and then -n is the name of the run. So every file that it spits out is not prepended, it's appended with whatever you type in here. So we'll type in something like test. Hit enter, and you can see that it did that very, very quickly. What it did was, if we scroll up a little bit, it gives you some information about who made RaxML, the version that we've used, how many threads we've used. So I mentioned there's different types. There's one called RaxML HPCP threads. If you use more threads, it can be faster. It tells you how many alignment patterns there are, how many gaps there are. All this is just extra information you don't really need. This is a repeat of what we've given it, so you can debug if something goes wrong. You can see exactly what you typed in to get there. This tells you how many seconds it took per bootstrap. This, you can read through it in your own time. It gives you all sorts of stuff. This is the important thing. So it tells you where it's printed all of your different trees. So because we've done all-in-one, we've made trees, we've made lots of different bootstraps. It's found the most likely tree, and then it's put the bootstraps on the tree. So you can do that in four separate steps, or you can do it all in one go, which is what we've done. So the one that you're really interested in is the one called bipartitions at the nodes. So if we go ls, you can see because I called it tests, they'll have test at the end. The one that we want is RaxML bipartitions.test. That's the one that appends the bootstraps to the nodes, which is the most correct. So coming on to the last bit, we need something to view the tree in. We always recommend Figtree, but if you want an online solution, there's something called ITOL, which is Interactive Tree of Life. It's very nice, shiny, pointy, clicky sort of thing. But we can install this doing sudo apt install Figtree, hit enter. You can see it's asking for my permission to continue because it wants to install some other things. It wants to upgrade some packages. Just type yes or Y, hit enter. It will get to work in the background. Some of these things take a long time to install, some are not so long. Depends on the speed of your internet connection as well, because it's downloading each of these files from the repository. Okay, so it downloaded all the files and now it's unpacking and installing them. Okay, now you can see it's finished. So we open this in the same way that we opened AliView. So you can just type in Figtree to make sure that it worked, hit enter. It will load the Java and some of the modules in the background, and you can see we have this point and click interface here. So what we do is we go File, Open. Everything is in a Downloads folder, which is Not immediately obvious to me where it is. Downloads, there it is. Search by modified. It's an example of sequences. Okay, so you have bipartitions branch labels, which is less correct because it puts the bootstraps on the branches. You want to go to this one here, which is called Raxamol bipartitions, and then underscore whatever you typed in. So open up the bipartitions one, and here you want to type in bootstraps, so you can find it afterwards. Hit OK, and now you can see this is the tree that we've made. So there's a few different things that you can do to manipulate the trees in Figtree. I always reroute it immediately to my outgroup, and in this case my outgroup was the reference sequence HXB2. This is the accession number, it's called KO3 something whatever. So you click on the node and then you just click up here to reroute, and it will reroute your tree. You then want to actually show the bootstrap values by going down here to node labels, open up node labels and click on bootstraps, and you can see each of these values now. So remember, the most correct way of interpreting this is looking at the nodes. So in 100% of bootstraps that it did, this node was in this position, in 50% it was in this position, in 90% it was in this position. Make it look a bit more pretty, you can go along the left-hand side here, you can go trees, you can order the nodes by increasing or decreasing evolution. So you can see that, you know, this sequence might have evolved from this sequence, might have evolved from this sequence. You can do, you know, different colors. So you can look up here, go node, clade, or taxa. I can change this by clicking on taxa and then going color, and then, I don't know, making it green, why not? You can play around, you can choose different colors and things like that. You might also want to look for drug resistance mutations, which is something that I do all the time. You can do this by using this online server. We use the Stanford HIV database. You can have a look at this link, some of you might know it already. You give it the consensus FASTA files, you can't give it BAM files, and it gives you resistance mutations. So it looks like this. So you go to, when you go to the website, it looks like this. You put in your sequences here, you press OK or something, and then it will give you this nice little report. It tells you the different resistant mutations, and then a list of drugs, what could be resistant, what might be susceptible, or whatever. There's some other tools. If you're not particularly happy with doing codey bioinformatics-y stuff, then you can use an online system called Galaxy. You can find the link here, which gives you loads of information. Essentially, what you can do is you can upload raw reads, and it will run a lot of these tools that we've used already. It can be a bit difficult to learn. You have to make an account and then upload the reads and then figure out which tools you want to use, and then you point and click and do some stuff. That runs on their server though, so it means that, you know, you don't have to worry about power being interrupted, because I mean, if you're running any of this on your laptop and power dies, your battery dies, it's just going to kill the job. You're going to lose what you've done so far. There's another tool which is really, really user-friendly. It's called Genius. It's very expensive, unfortunately. It's $450 per person per year, but it's very user-friendly. You can do all sorts of things with it. I'll show you an example because I'm still sharing my screen. Let me open Genius. We'll have a look quickly. So, I mean, you can do everything in here. You give it the raw reads, you can make an alignment, you can build BAM files, you can even make trees in here. So here's a BAM file that I made. You put everything into here. It's got a nice viewer. It shows you all the reads down here. You can do sorting. It gives you loads of metrics. You can make trees up here with lots of different tree building software. Genius is nice. There's a 14-day free trial. I'm happy to show you guys how it works if you want to learn. Besides that, I mentioned earlier about being accredited by PANGEA. Lucy's asked me to ask you, if you don't want to make BAM files, if you don't want to make consensus sequences yourself, we've made all of them already. They're available at Oxford. You just need to be accredited. So there was an email that went around in March, February or March this year, with an accreditation form. Lucy is going to send around an email immediately after this for people that want to get accredited. There is funding for you to do the course which is required to get accredited. We recommend one by city. It's $129 or something. It's valid for so many years. Once you've done that and you've got a certificate, you then just fill in the sort of letter of intent and say what you want to do with the information, the data, sorry, and then we'll send it to you and give you any support that you need to do analyses. Email Lucy if you need help with that. Email me. I can help you. Otherwise, I guess that's pretty much it. This is only part one of sort of the basic introduction. We're going to have another one on the 14th of November, where hopefully, if you want to use any specific software or you need more help with doing any of the analyses, you can email me in advance and I will build that into a PowerPoint.